from the Howard Hughes Medical Institute. The 1995 Holiday Lectures on Science. This year's lectures on the double life of RNA will be presented by Dr. Thomas R. Cech, Howard Hughes Medical Institute investigator, distinguished professor at the University of Colorado Boulder, and 1989 Nobel laureate in chemistry. The first lecture will discuss catalysis, chemical and biochemical. And now, to introduce our program, the president of the Howard Hughes Medical Institute, Dr. Pernell W. Chopin.
Good morning. It's a pleasure to welcome all of you to the 1995 Holiday Lectures on Science. And we want to welcome not only all of you here in our auditorium in Chevy Chase, but those of you who are watching on television in thousands of schools across the country. You're in for a real treat. Our speaker today is Dr. Tom Cech, who won the Nobel Prize in Chemistry in 1989 for his work on RNA catalysis. But he's also a great teacher, and you're going to hear firsthand about the process of scientific discovery and about some of the most exciting things that are going on in research today. I know that you'll find his talks interesting, and I hope that at least some of you will consider pursuing careers in science. I spent almost all of my own career at doing work at the bench, and I can tell you that there's nothing more fascinating, no more rewarding for society than searching for the biological secrets that underlie health and disease. Science holds the key to cures for diseases, cancer, heart disease, mental illness, and scores of other important things that plague us. We need to know the answers to such things as why tumors grow out of control and why the AIDS virus overwhelms the immune system. To do that, we must ask questions first at the basic level, questions about genetics and cell biology and other areas of basic research. This research may not focus initially directly on specific diseases, but it lays the intellectual background from which ultimately new cures and methods of prevention of disease will flow. It also opens up a whole new view of the world around us, and it's a wonderful time to be a scientist. Dr. Cech and his other colleagues at the Howard Hughes Medical Institute are exploring the frontiers of biology every day. Our institute is the nation's largest private philanthropy. It's a medical research organization. It has scientists supported by excellent research teams that work in 62 locations across the country. There are about 280 of those scientists. They work in laboratories from Boston to Seattle, from Birmingham to San Diego. There are several Nobel Prize winners among them and a great many very distinguished scientists. Now, in addition to research, the Howard Hughes Medical Institute is committed to enhancing science education, to training of tomorrow's scientists, young people like you. It has the largest private grants program ever mounted in U.S. history to promote science education. We sponsor hundreds of science education projects in universities, in museums, in medical schools, and other institutions across the country. And this holiday lecture series is just one part of our effort to train the Tom Cechs of the 21st century. Dr. Cech received his bachelor's degree in chemistry from Grinnell College in Iowa and his Ph.D. from the University of California in Berkeley. He then did postdoctoral work at the Massachusetts Institute of Technology. So whether you're watching this from the Midwest, from the West Coast or the East Coast, you all can rightfully claim Tom as one of your own. And that's particularly true if you're watching from the Mountain States, because Dr. Cech is a distinguished professor of chemistry at the University of Colorado in Boulder, in addition to being an investigator of the Howard Hughes Medical Institute. The title of his series of talks is The Double Life of RNA. The first lecture is on catalysis, chemical and biochemical. We're first going to show you a brief video about Dr. Cech, and then he will speak. So on behalf of the entire Howard Hughes Medical Institute community, I welcome you all to the first in our 1995 Holiday Lectures for Science. Tom, we'll run the video first, and then I'll turn all these bright young minds over to you. Thank you.
I made the decision to be a scientist at such an early age, I have trouble remembering. My parents, who have saved absolutely everything that I ever wrote in grade school and on, have documentation from the time that I was in probably second grade. What do you want to be when you grow up? I'm going to be a scientist. The question of why did I decide to specialize in biochemistry involves a lot of serendipity. The reason I decided to major in chemistry was because I took an exam to pass out of freshman chemistry entirely and didn't succeed in passing out of it. And I was so mad, I said, I'm going to learn this stuff because it seems like material that would be worth knowing. It wasn't until my senior year that I took one semester of freshman biology and learned about molecular biology, which I really had very little experience with before that at all. The whole concept of everything that goes on in a cell as being chemical reactions, but chemical reactions that are controlled, regulated, manipulated for the purpose of cellular growth and cellular response is a fascinating area that man has pondered for thousands of years. A lot of the questions that people have been asking for thousands of years are being answered, and it's a very exciting time to be a biochemist or a molecular biologist. A lot of people in the general community think that the medical implications of the research are, of course, what must be driving all of us. And we do pay attention to the possible medical spin-offs, but the real honest truth is that what drives us is curiosity about how nature works. And in the short term, we follow our nose a lot. And that turns out to be a very successful way of carrying out science. You develop a sort of intuition, and just following your nose can lead to discovery of things that are in some cases far more significant than the straight path towards the goal that you were originally sought. The sort of dry explanations of science that you read in a textbook is not the way that science is done. Science is done not by a scientist sitting down and saying, well, here's the scientific method. We have to have our hypothesis, and then we, this sort of set list of ways that you go about proving something, but that in fact scientists work in a much more haphazard, much more interesting way. So my goal for the holiday lectures is to bring to life the scientific process and give students a glimpse of what it's really like to be a practicing scientist. Good morning, and welcome to the Double Life of RNA. The sort of science that I'm engaged in is called molecular biology. So I think it might be a good place to start to ask the question, what is molecular biology? Well, it is not the sort of biology where one studies the social behavior of hyenas, although that's a fascinating area also. But instead, a molecular biologist is interested in the molecules inside of a living cell. What do they look like? What are their structures? How do they interact with each other, and in some cases actually react with each other? How are they regulated, and how do these cellular molecules promote cellular growth, cell division, cell movement, responsive cell to its environment, and also how do these molecules promote the interaction of cells with each other, for example, to build a tissue or an organ. So given that I'm interested in molecular biology, you might be wondering why these chemistry demonstrations are set up on the stage with the beakers and the flask boiling. What does that have to do with molecular biology? So I'd like to ask you, does somebody in the audience have an idea of the relationship between chemistry to molecular biology? Please, don't be shy. Yes, in the back.
Same chemical processes take place inside the cell as test tube or beaker. Can you illustrate that?
Right, so very good. So the answer is that a lot of the reactions that take place inside of the cell are in fact just chemistry, and they involve chemical compounds, and so we can illustrate them on the benchtop. Any other reason for using these sorts of demonstrations? Yes?
Isn't chemistry considered the basis of all life processes? And so in demonstrating molecular biology, we would need to use chemistry.
Excellent. So chemistry is the basis of all of the life properties and processes. Anyone else? Why do the demonstrations out here on this table, why don't we do the actual biochemical demonstrations? Does someone have an answer to that? In the back there.
Is it because it's at a much smaller scale?
Excellent answer, and please speak up when you give the answers. She said that it's because it's on a much smaller scale. And so if we were restricted to biochemical demonstrations, it would be hard to get enough material within our budget at least, in order to show a whole beaker full of biological material undergoing a reaction, whereas simple chemical reactions can illustrate many of the same principles in a way that we'll then later try to relate to the biochemical example. So let's start out with something very simple that has to do with these balloons, and there's a third one that's fallen onto the floor. And I'm going to use this just as a type of an analogy to illustrate how scientists approach problems, and then we'll make the biochemical correspondence a little bit later. So I'm going to tell you to start with that these three balloons are each filled with a different gas. One is filled with air, one has helium in it, and the other has hydrogen. So do any of you want to make a hypothesis or hazard a guess as to which of these balloons might have which gas in it? Yes, back there.
The one that's on the table is probably air.
Okay, this is probably air, and then
The orange would be hydrogen and the yellow would be helium.
now, okay, so that's an interesting guess. So I need to know your reasoning, though. How did you differentiate between them?
Well, in my recent experiences, I've seen that when you blow up a balloon with your mouth, over a period of time, it starts to like sort of fall down. It starts to lose its, I don't know, its lift.
Okay, so you're talking about its lift has to do with the density of the gas inside the balloon relative to what? The density of the air, because that's what the balloon is in. So I think you've correctly surmised that this is the one that has to be holding air, and that the other two have to be holding the hydrogen and the helium. But does someone else have an idea? And you made a guess, but I'd like someone else to comment on how we might be able to differentiate the hydrogen from the helium. Over here.
Well, I think the yellow one has hydrogen in it because it seems to be pulling more on the string. It's less dense than helium.
Okay, so a comment was made that perhaps the yellow one is the hydrogen. You said that the orange one was the hydrogen, so we have so far one vote for each. But the comment was that if we could make, or let me paraphrase your comment by saying that if we could make a careful measurement of the density of the gas, since hydrogen is less dense than helium, that would be an experiment that we could do to differentiate the two. Anybody else have a different idea? Because a scientist doesn't just look at things and make up his mind or her mind. A scientist interrogates the system. Isn't that a great word? But you have to do an experiment. You have to probe the balloons in some way to really answer this question. Back there.
Well, hydrogen is flammable and helium isn't.
Hydrogen is flammable. Well, that's a great idea. Well, I just happen to have a device, a very simple device that will allow us to make a test. I need 2 volunteers from the audience. So if you can come down from the middle there, and how about you in the second row? Yes. And if you can each put on some safety goggles. And you're Katie? And you are Erika. Okay, Erika, I'm going to have you since you're over on the left, start out, and I would like you to perform this interrogation of the yellow balloon. And now just wait a minute here. Let me get, this is very safe, I assure you, but do it at arm's length and don't be shy, and go right for the yellow part of the balloon. No, don't jump, just simply hold the candle up to the balloon itself. No, you have to be a little more bold here. And we'll see. Don't break the string, but get the balloon itself. Reek, I'm going to have to ask for another volunteer. Okay, anyone? What's the answer? Hydrogen or helium? Helium. Okay, so the helium escaped, but it was not flammable. And so, Katie, that leaves it to you. Now, we can't be sure I was telling the truth. They could both contain helium, or perhaps there's even another gas that's lighter than air. Okay, now let's be prepared here, just in case. Little higher. So that's a simple demonstration then of the scientific method. Let's give a hand to Katie and Erika for helping out. Now you might wonder whether there was any toxic gas released in that experiment, and as you can see from the slide, we're really just producing water. This is an example of an extremely simple reaction in which two hydrogen molecules, each of which contain hydrogen atoms bonded together, react with an oxygen molecule. These bonds are broken, and new bonds are formed between oxygen and hydrogen to give two water molecules as the product of this reaction. So even though this is a very simple case, biochemical reactions are like this as well. They involve not the destruction of individual atoms, but the rearrangement of atoms to form new molecules. And so there's a lot of similarity. I think, though, that it might be worth asking the question, how is this sort of a simple reaction, chemical reaction, different from biochemical reactions that take place in the cells of your own body? So does one of you want to give an answer to that question? Yes, in the front.
Because like in the cellular catalysis, all those enzymes accelerate the reaction.
So the answer was that in the cell, the reactions have to be catalyzed, and they have to be controlled by enzymes. They don't just happen when you light a match, or at least that's not the kind of catalysis that is going to be involved for a cellular reaction. And that's one excellent answer. Does someone else have another answer to that question? The difference between this sort of biochemical reaction. Let's think in terms of, well, again, we talked about the scale of the reaction before. Obviously, the reactions are taking place on a much smaller scale. One of the products of this reaction that isn't listed on the slide is heat. And you could see, maybe you could, if you were in the front row, maybe you could even feel the heat that was generated. Do you think that's a similarity or a difference? Do you think that biochemical reactions sometimes generate heat as well? Yes.
Yes, but not on the same magnitude.
Not on the same magnitude, that's right, but they do generate heat. They don't generate heat on the same magnitude because, of course, we have much less material involved, and so the heat is going to be proportional to the amount of material. But one of the reasons that you, as a mammal, are maintaining a 37 degrees C as a body temperature has to do with the heat that's being produced by metabolic reactions inside of your body. I would say that another difference between the demonstration that we did and the biochemical reactions that wasn't mentioned is that you could see that was an explosion. It happened all at once. It happened very quickly, and then it was over. Well, inside of living cells, it's important for reactions to occur on a more continuous time scale, not in an explosive manner, which might be cited as another difference. So let's move now from the realm of chemistry to some of these biochemical reactions. And I would like to start out by just reminding you of something that you all know, which is that in living cells, information is stored in the form of deoxyribonucleic acid, the famous double helix of DNA. And when I talk about information, I mean information that tells a cell how to build a particular protein, shown over here on the right. And how is this information that's stored in the chromosomal DNA eventually used to design a particular protein? Well, the DNA is first copied into a chemically rather similar molecule called ribonucleic acid, or RNA, and then groups of 3 of the building blocks of RNA. And as you know, these building blocks come in 4 what I like to call flavors. There's an A, a G, a C, and a U in the RNA. And different triplets encode a particular amino acid. For example, A followed by U followed by G encodes a methionine, 3 U's in a row encodes a phenylalanine, etc. And so the order of the building blocks, these individual units along the RNA molecule, as copied directly from the DNA molecule, determines the order of amino acids that are laid down into this chain of protein. And the order of the amino acids, in turn, determines the way that the protein folds up, which will determine the function that the protein has. For example, a protein might be the protein pepsin, which is an enzyme in your stomach that's digesting your breakfast right now. And that pepsin has a particular gene that specifies production of that protein. Or myosin is another protein that's involved in muscle movement. The myosin protein will have a particular stretch of DNA that encodes or specifies the construction of the myosin protein. Now, a simple everyday analogy would be that the DNA is like an archival master copy of your favorite videotape. So you've got one of those masters, and from that you can copy a limitless number of copies of the tape, which in terms of their information have the same information as the master copy. That's the RNA equivalent. Then you need some kind of a machine to plug the videotape into, and that's a VCR machine, which is analogous to the cell's ribosome, which is its protein synthetic machinery, where the amino acids are lined up with the triplet codons along the RNA using this transfer RNA intermediary. And then finally, you have the final output of this series of events, which is, in the terms of a videotape, an image on the television monitor. And that is equivalent then to the functional protein that's produced from this chain of events in the cell. Now, over the next few lectures, we're going to have a slightly less even view of these 3 molecules involved in information transfer. So you can see RNA is now in the big green letters, because RNA, like DNA, has the ability to carry and store biological information. But what's been found more recently is that RNA shares with protein the ability to be an enzyme or to catalyze biochemical reactions. And it's this dual life or double life of RNA that's going to be the topic of the lectures coming up. I think it's very useful to get a picture of what these molecules look like, because their function and structure are intimately connected. And so let's start with looking at a picture of a protein, in this case a protein which catalyzes a reaction on a small sugar molecule. This is first of all, you can see in terms of the number of atoms involved, it's huge compared to the oxygen and the hydrogen that we used in the first chemical demonstration. It's a large globular molecule. This one has a molecular weight of many tens of thousands rather than just a molecular weight of under 100. It has an active site cleft, which envelops a small sugar molecule in this case. And then there are chemical groups provided by the protein which in a way, encourage the small molecule to undergo a very specific sort of chemical transformation, and that's the catalytic step. So this is a protein. Now, if you ask the question, well, could a nucleic acid, is it reasonable that a nucleic acid could be a catalyst as well? If you start out with the type of nucleic acid that you're used to thinking about, the one that is highlighted the most in your high school biology texts, you think about the double helix of DNA, and it seems to have a very different sort of structure. It's not globular at all. You can see that even from this short stretch of DNA, but this amount of DNA would not be enough to encode a protein. A gene at this scale would have to extend clear off the screen, wrap all the way around this auditorium in order to be long enough to have enough information to specify the production of a protein. So this molecule, instead of being a glob, is much more like spaghetti. It's a long, thin molecule. And in fact, I'm reminded of the videotape analogy again. This is the cell's videotape, and like a videotape, it's a linear, rather regularly structured molecule, very well suited for storage of biological information. Now, when we move from DNA to RNA, we move from a double-stranded molecule to a molecule that has a single nucleic acid chain, but that single chain can be folded back on itself, as shown here in the case of transfer RNA, to form helical regions, but also more complex structures, such as a very intricately folded region here, a hole in the molecule, an indentation at this point, a place where the chain turns around and forms a loop. Down at the bottom, a single-stranded extension over on this end, which carries the amino acid. And so RNA, although single-stranded, isn't necessarily structureless. In fact, we think of RNA quite the opposite, that the RNA takes on a variety of structures that are critical to its biological function. And finally, when we move from transfer RNA to a type of RNA molecule which is known to have catalytic activity, we see a structure that has quite a few helical regions packed together in a very specific way. And this molecule is shown here as a framework so that you can peer inside of the helices and see the overall architecture of the molecule. But if we looked at this, as we will later, in the form of a space-filling model where you can see all of the atoms, you would have trouble from where you're sitting telling the difference between this catalytic RNA and the globular shape of the protein enzyme that I showed you earlier. And so let's now continue comparing and contrasting chemical and biochemical catalysis by doing another demonstration. This one, which I like to call Mr. Lincoln Glows, the Mr. Lincoln here is a Lincoln penny, which of course is made out of copper. And what we have in the beaker is some acetone, which is a simple organic compound. And just so that I'm careful to follow my own safety rules here, I'll put on the safety goggles. And the acetone can undergo a decomposition reaction. It can break down, but this happens extremely slowly under normal circumstances. So that acetone, in fact, would all evaporate into the air of the auditorium long before we would get any significant decomposition, chemical breakage of the acetone molecules. But something quite different happens in the presence of a hot copper disk, in this case provided by Mr. Lincoln. So I'm going to clamp this close to the surface of the acetone. And if we could have the lights down a bit, I think you can see, and maybe you can, if we could have the lights quite far down so that the people in the audience can see the penny directly. Because I think if we turn off these lights, you'll actually, even from the back of the auditorium, be able to see the penny glowing. Is it possible to go completely out on the lights? Now, we tried this last night, and although we don't have an hour to sit and stare at this, I will tell you that that penny will continue to glow that orange hot color for an entire hour as the acetone is decomposed. So let's look then at the reaction that's involved. If we could now have the video off and go back to the slides. In the presence of this metallic copper surface, the acetone molecules land on the copper surface, and that speeds up their breaking apart into two other organic molecules, this ketene and methane. Methane you know about. This is just heating gas that's used if you have a forced air furnace in your house. This is natural gas. And then in the presence of oxygen from the air, a combustion reaction takes place where these small organic molecules burn to produce carbon dioxide and water, and of course heat, which in turn keeps the copper catalyst hot, which allows it to continue participating in the breaking down of the acetone. So that's one type of chemical catalysis that we can use as a model system for biochemical catalysis, the providing of a surface that has the right spacing of copper atoms to encourage the molecules of, in this case, acetone, to decompose in a very specific way. The so let's think about this now in terms of the principles of catalysis, and I have a few of them mentioned on the board here. First of all, a catalyst is something that speeds up a chemical reaction, or if it's a biochemical catalyst, it speeds up a chemical reaction that's important for a living cell. It does that not simply by speeding up the reaction in the way that it would occur uncatalyzed, but by providing a new pathway for the reaction to occur. In the case of Mr. Lincoln and the decomposition of the acetone, it was by providing a surface that the molecules of acetone could sit down with and their chemical bonds be destabilized in a way that led to decomposition. The catalyst participates only transiently in the reaction. It's restored in its original form afterwards, and this can also be seen with the copper catalyst, because if we look at this penny after it's through with the reaction, we'll notice that the copper is still there. There's been no decomposition of the penny. It's not been eaten away at its edges. If you were to weigh it before and after, you'd see that the copper content was still there. Again, providing a demonstration that the catalyst is participating only transiently. I'd like to do another demonstration, a little bit more dramatic one, of catalysis that involves this time soluble metal ions rather than a metallic surface. And this next demonstration has to do with the oxidation of tartrate into an oxalate product. And before I show you that reaction on the board, I think we should do the demonstration. And at this point, I would like to have, again, a couple of volunteers, somebody who's close to the, could you come up? And yes, please. There, second from the end. And if you would put on, again, we always like to stress safety here, so if you would put on a lab coat. We have Sarah and Rizwan. Okay, and we're going to start out now by just pouring this. Oh, no. No, is correct. Thank you. We're not going to pour that in. We're going to add hydrogen peroxide. And again, if you could put, we forgot the safety goggles here. We have to all be appropriately dressed for this reaction. So Sarah, if you could pour the hydrogen peroxide into that beaker and just put the entire contents into the tartrate. Just pour it in, and there's a stir bar there, and you can give it a little stir. And this oxidation reaction is accompanied by the evolution of a gas. Now, what do you see, Sarah? Tell us, what's your observation of the system?
It's steaming.
Is there a very vigorous reaction? No, very little is happening. The steam is because we started out with a hot solution. So what we need to speed this reaction up so that we don't have to all sit here for several hours waiting for this oxidation to take place is a what? Catalyst. So Rizwan, why don't you add the catalyst, which is that copper chloride solution. And again, you can just dump that directly in. You can see, note the color. It's a pretty pink color. And now this evolution of the gas, which is accompanying the oxidation reaction, is quite visible. And what else do you notice? What happened to the color?
It's changed.
It's changed from pink to what would you call that?
Kind of dark brown.
Yeah, brownish, or almost, I think it has a little bit of a greenish tinge. The reaction rate has been accelerated so much that we can be patient enough to actually wait for the decomposition to be complete. And what's happening to the color now?
It's turned red.
It's turned back to that same sort of burgundy or dark pink color that the catalyst had originally. Okay, very good. If you, let's give Sarah and Rizwan a hand. Let's look then at the chemistry that took place. The tartrate molecule, you can see, has 4 carbon atoms decorated with oxygens at various positions. And although I'm not going to talk about the mechanism of this oxidation that takes place in the presence of hydrogen peroxide in detail, I think you can see that if you would take this molecule and cut it down the middle, you would have 2 products, each of which would contain 2 carbon atoms. And so, in fact, it is that carbon-carbon bond that is broken during this oxidation reaction. The other products of the reaction are molecular oxygen and carbon dioxide, which accounts for the bubbling that we saw in the solution. And this is a very nice, the color change gives a beautiful illustration of one of the principles of catalysis, which has to do with this transient participation and then restoration of the catalyst in its original form at the end of the reaction. So the cobalt metal ions in solution, are in solution, and the tartrate, which you can see has a negative charge. The cobalt ion would have a positive charge. They interact with each other. The tartrate wraps itself around the cobalt ion in such a way that it is subject to attack by this species produced by this hydrogen peroxide, resulting in its breakdown into these molecules of oxalate. But then the products can be released from the cobalt ion. And in the cobalt ion, when it's tied up with the tartrate, changes its color to that brownish, greenish color. And then when the products are released again, the ions are back in the free cobalt ion form in solution. And then you see the pink color restored, which is then evidence, visual evidence, that the catalyst has not been consumed, but is restored in its original form. And if we had enough time, we could use that pink solution over and over again to keep this, to demonstrate that the catalyst has not been chemically changed and that it can be recycled an endless number of times. Now, for the final moments of the presentation, let's go back now from chemistry and re-enter the world of biochemistry and relate catalysis again to an RNA system. And this is a very small catalytic RNA shown in the upper left-hand corner as this sort of steely blue color. This is the RNA molecule that can act as a catalyst, but it doesn't have any molecule to interact with in the upper left-hand corner. However, it can bind another strand of RNA shown in pink, which is the one that's going to actually undergo chemical change. We call the pink RNA a substrate RNA. And once this complex is formed, in the presence again of a metal ion, so many biological catalysts, including these RNA catalysts, use metal ions. In this case, not cobalt metal ion, but perhaps magnesium or manganese ion, magnesium being a very common +2 charge metal ion that's very common inside of living cells. In the presence of that metal ion, a chemical reaction takes place, the details of which are shown here, that results in what the artist has shown as a little explosion, a demolition of this pink RNA strand at a very specific place. So the single RNA chain is broken into two product chains. Those cleaved products can be released, restoring the catalyst in its original form, and this catalyst can then interact with the second substrate RNA and go around this cycle perhaps hundreds of times in an hour, and many other biological catalysts have a faster turnover number than that even. But the main point in terms of the principle is that the RNA catalyst is not changed in the process and can go through the cycle an endless number of times. So to recap what we've covered in the first lecture, we've decided, and you helped me decide, that the chemical reactions that we can demonstrate on a benchtop, although there are some differences in terms of scale and in terms of perhaps spontaneity and explosiveness compared to biochemical reactions, that there are a lot of similarities between chemical reactions and biochemical reactions, and that biochemical reactions which are catalyzed by enzymes, which can be either protein in nature or they could be RNA catalysts, operate according to principles that are very similar to the way that metal ions or a metal surface, in the case of the Lincoln penny, would catalyze a chemical reaction. Thank you. And I've been told that we have some time for questions from you, so please speak up, speak loudly. Yes, in the middle.
What exactly? You're a molecular biologist, are you?
Am I a molecular biologist? Yes.
Okay. Because see, that's what I want to be. And what exactly, motivated you besides what you said before to really go into that field? Curiosity or just...
So the question was, what motivated me to go into molecular biology as opposed to some other sort of field? All of my training had been in chemistry, and in particular in physical chemistry, which involves the interaction and reaction of molecules in the gas phase and a lot of spectroscopic interrogation of the reactions in the gas phase. And interestingly, it was really my temperament which drove me away from that field and into molecular biology, more than fascination with the scientific material. In biochemistry or molecular biology, you do an experiment and get an answer back from the experiment almost on a daily basis. Very quick. You get an idea, you can test it, you get an answer. Sometimes the answer is that you don't get the answer that you were hoping for, and you have to rethink the experiment. But there's this constant interplay between ideas, experimentation, back and forth. In many areas of non-biological chemistry, people spend months, even years, building a machine to test a particular idea. And I'm too impatient to wait around that long. So that was part of my motivation. Another question back here.
Yeah, what made you decide to research RNA and specifically its catalyzing properties?
So the question was, why did we decide to research the catalysis by RNA? And the answer was that we didn't seek it out. It sought us out and found us, and you'll hear that story in the next lecture. Next door, yes.
This may also be addressed in the next lecture, I'm not sure. Are there any specific examples we might be familiar with of RNA acting as a catalyst, biological catalyst?
So what is an everyday example of RNA acting as a biological catalyst? I think the most important example in living cells has to do with the process of protein synthesis. The ribosome has been known for decades to consist of 3 or 4 RNA molecules, but not just RNA. They are associated with a myriad of proteins as well, very specific proteins. So it hasn't been easy to ask the question, is it the protein parts that are the enzyme factors and the RNA is just sort of serving as a scaffold holding things together? Or is it the other way around? Is it the RNA that's the active component and the proteins are helping modulate that RNA activity? Current opinion has swung in the direction of the ribosome being an RNA catalyst, in part due to some wonderful work that's been done in Harry Knoller's laboratory at the University of California, Santa Cruz. The final answer to that is, I'm sure, going to be that the ribosome works as a ribonucleoprotein particle. In other words, obviously both the RNA and the protein components are essential, but at its very heart, the catalysis of transfer of an amino acid to a growing peptide chain appears to be an RNA catalyzed reaction. So that's, I think, the one example that is the most sweeping and the most central to biology. We'll talk about others later. Other additional questions? The northwest corner has been very quiet. Does someone up there have a... Yes, down here.
What are the drawbacks of doing research in molecular biology?
What are the drawbacks? Do you want to expand on that? What do you have in mind?
Well, are there any disadvantages? Like, was the long time you have to put into? Are there a lot of mistakes? Anything that you don't like about it?
What are the drawbacks to engaging in molecular biology research? Sure, there are long periods of time of frustration. When experiments aren't working, and especially if you're trying to understand things at the very fringes of knowledge, where no one has tread before, which is the most exciting place to be operating. It's also the place with the least certainty. If you repeat someone else's experiments in the laboratory, you can probably make progress very steadily. On the other hand, no one's going to be very excited about the results. So if you're operating in totally new frontier territory, there are no guarantees about how long it will take to understand the system that you're trying to understand. And certainly persistence is required, and also flexibility, that one has to, if your first approach to a question doesn't bear fruit, you have to be flexible enough to do an end run and attack the problem from a different direction rather than continue hitting your head against the same wall all of the time. But I think those sorts of frustrations are not particular to molecular biology. I think that most of the sciences, that science in general has that sort of a drawback. And it takes a type of personality a person who can be simultaneously patient and impatient. You have to be willing to work for a long time to get to a result. So you have to have patience, but also you have to really want to get there, and so you have to be, at the same moment, an impatient person. Yes, down in front.
Why did you heat the sodium tartrate? Was it to increase the rate of reaction?
Yes, that's a good question. Why did we heat the sodium tartrate? It was at about 50 degrees Celsius when we started the reaction, and you're absolutely right. It has to do with the rate of the reaction. Many reactions will occur more rapidly at a higher temperature, and in this case we were able to speed up both the uncatalyzed and the catalyzed reaction. Now you may say you didn't see the uncatalyzed reaction speeding up. Well, we didn't have a sensitive way of monitoring the very small amount of oxygen and carbon dioxide that was being evolved in that reaction before we added the catalyst. But had we been able to do that, you would have seen that the uncatalyzed as well as the catalyzed reaction would speed up at the higher temperature. Additional question, yes?
What is the catalyst in the purification?
So the comment was that you've purified DNA in the laboratory, and you're interested in the catalysis of that. Well, during the purification, there isn't a catalyst during the purification, but the cell uses a catalyst to build the DNA to start with. And so that's called a DNA polymerase. A polymerase is an enzyme that strings together the individual, what we call monomer units, to make a polymer. In the case of DNA, the monomer units are the nucleotides, which are in the form of A, G, C, and T, are the 4 building blocks. And they are strung together by an enzyme called DNA polymerase using a pre-existing strand of DNA as the template that determines the order that the nucleotides are laid down. So the catalysis, in fact, involves not only the enzyme DNA polymerase, but also a pre-existing chain of deoxyribonucleic acid. In the back, in the gray sweater.
Since RNA is coded from DNA, wouldn't DNA then have catalytic properties if RNA is coded from it?
So that was a very perceptive question, that if RNA has catalytic properties, why not the DNA that encoded the RNA? What's so special about RNA? In fact, DNA in its single-stranded form has been recently shown to have catalytic activity. For example, Jerry Joyce at the Scripps Institute in Southern California has done some nice experiments along those lines. I think that the way that DNA normally occurs in the cell, locked into this very regular double helical structure, is in part something that has occurred in nature to prevent the DNA strands from folding up by themselves and performing interesting reactions such as catalysis. Because when you want a very secure stronghold, a very safe place to store information, you don't want reactions to be taking place in the genes themselves. And so by locking the DNA into the double helical structure, it's prevented, the 2 individual strands are prevented from folding up into shapes that by themselves might be able to perform catalysis. As you'll see in the next lecture, one type of catalysis can be self-catalysis, can be cleavage of a bond within the same molecule. And the last thing we would want our chromosomes to be doing would be for the strands to fold up and catalyze their own cleavage or rearrangement in an uncontrolled way. But you're absolutely right. From just from a chemical basis, RNA is not that different from DNA. It does have an addition, as you can tell from the name, deoxyribonucleic acid is missing what kind of an atom? An oxygen atom on every one of the building blocks. So RNA has an extra oxygen atom. It's actually in the form of a hydroxyl group an OH group. And that does give RNA additional reactivity and additional structural possibilities relative to DNA. But it's a fairly small difference. And so if RNA can be a good catalyst, you would expect DNA should at least be able to be a fair catalyst. Question over here.
This is kind of related to the last one, but is it that extra hydroxyl group that allows the RNA to form the different structures? And why do the different kinds of RNA form the different structures?
So the first question was, is it that extra hydroxyl group which allows the RNA to form structures? And the answer is that it's that is one feature of the RNA that allows it to form a very particular sort of a globular structure that can act as a catalyst, but other interactions, such as pairing of the bases, complementarity between A and U, G and C, also is another sort of interaction that's very much involved. And your second question was?
Why do the different kinds of RNA, like why does the TR, is that the arrangement of the nucleotides that makes
Exactly. So the question is, why do different RNAs that have different functions, how do they know how to fold up in a different way? And the answer is that what we call the primary structure, the first level of structure, which is the order of A's, G's, C's, and U's along the chain, which is, of course, determined by the DNA from which they were copied. That order determines the way that the RNA folds and pairs with itself. And if you change that order of A's, G's, C's, and U's, you will perturb, perhaps in a useful way for some other process, the way in which the RNA folds. Thank you very much for your excellent questions. I'm sure we'll have time for some more later.
Thank you, Tom, for a wonderful first lecture. We're going to take about a 30-minute break, and then we will come back for the second and final lecture of today. Tom will take up where he left off and tell us more about his research on catalytic RNA. The title of his second lecture will be RNA as an Enzyme, Discovery, Origins of Life, and Medical Possibilities.